Vanakkam. Welcome to this course on classics in neuroscience. We started looking at the physiology of the action potential, or how nerve impulses travel through a neuron. In this video, we will look at how nerve impulses were being recorded. First records of nerve impulses or neuronal action potentials, and how to record single action potentials. In the 19th century, even in the late 19th century, sensitive equipment to measure action potentials were not available. It was found that if we stimulate a muscle nerve, that is a neuron attached to a muscle, if you electrically stimulate that, the muscle twitches. So, electrical stimulation of the neuron causes a mechanical action in the muscle. This suggested that perhaps muscles also have a conducting mechanism that is similar to nerve impulse conduction that happens in neurons, or maybe muscles are also similar to the neurons in the sense that they may also be excitable, they may also conduct action potential. Was the insight that we could come from this. When you stimulate a neuron, the muscle will twitch. Other than this, it was not clear the relationship, input output relationship, how much stimulation leads to what kind of response. These kind of things were not measurable in the 19th century. Then of course, the string galvanometer was developed. An early pioneer in this field was Keith Lucas. I mentioned about the string galvanometer and how super sensitive it was. On the one hand, we did not have sensitive equipment. On the other hand, we had equipment like string galvanometers that were very sensitive to small vibrations. Because string galvanometers were super sensitive to small vibrations, it was found that heavy tables were needed to prevent artifacts or unwanted vibrations or unwanted measurements of the string galvanometer. Keith Lucas used to have experimental tables that were weighing many tons. So, Lucas developed many innovative experimental apparatus, many of which today would look very funny, but in the good old days this is all that was available. So, he had this massive brass arcs. So, he had these massive brass arcs through which he swung a pendulum to trigger or to produce triggers at specific points in time. Stimuli and muscle responses were recorded on smoked drums, and electrical recordings were made on films. So, this constituted the electromechanical era of recording, electromechanical era, not the electrical era. Today we record this in the computer. So, in the good old days it was recorded using some electromechanical apparatuses. Electromechanical era of physiological instrumentation. We performed all these experiments mostly on the muscle nerve of frogs. For a long time, this science was called as the frog science. The frogs were the main experimental animal models. It was assumed that the study of the frog muscles would lead us to understand bigger animals, but also study of the frog muscle would help us to understand the brain itself. So, this is a key strategy that is frequently used in neuroscience, that is you use the model system that is smallest, that can answer the question that you are asking, but not bigger. But the results from this model system must be generalizable to bigger systems, is a key critical component. Of course, because of this, because it is too narrow, sometimes it has its own value, but sometimes it becomes too restrictive to operate with these kind of constraints. So, action potential recording in the good old days used to happen using extracellular recordings. Although this is not as accurate as intracellular recordings, this was the mainstay, this was the main method of inquiry in the early 1900s. the pioneer was of course Keith Lucas, but unfortunately during a factory accident in 1917, although he was a scientist, during the First World War he was working in a factory. Unfortunately in an accident he passed away. So his lab was taken over by one of his students, Adrian. Adrian continued the legacy of Keith Lucas, and Adrian and his students performed a series of studies that improved our understanding of the nerve impulse and the conduction. So Adrian and his students used string galvanometer to study how afferent impulses, afferent means sensory, efferent with an E means motor. In this case, afferent impulses from receptors in frogs muscle. What are these receptors? Muscle spindles. These are sensors within the muscles that are sensitive to stretch. So they recorded with this capillary electrometer and 3-stage amplifier, of course not the electronic amplifier that we have today, the early amplifiers. And they stretched the muscle with varying weights. So the amount of stretch varies. If the stretch varies, it turns out that muscle spindles activation will vary. The exact mechanism of how muscle spindles work, I will share a video with you. Muscle spindles video. Beyond the scope of the current discussion, those who are interested can check that video from the neuroscience of human movements course. It turns out that as the length of the stretch increases, the output from the muscle spindle increases. So the frequency of firing increases, and they found that frequency increases with load and decreases over time, and they found that there are about 5 to 100 impulses per second. So the good old days, these were fantastic findings, and there was also some rhythm that they could find. This was perhaps due to the refractory periods and return of excitability. So after the refractory period, the system, the neuron comes back to a position in which it can again be excited. So duration of excitation, amount of excitation, both of these have an impact on the output. They found that the information, depending on the duration of excitation, the amount of excitation, the frequency of firing and the number of impulses per unit time changed. So there was this encoding of information in this action potential, in these trains of action potential. This was perhaps the first proper description of this phenomenon. This started a whole new field of study. A lot of papers and books and material, research material came following this. Described in the book by Adrian, the basis of sensation. So what's shown here in this picture is the loading of these specific muscles, sciatic muscle or gastrocnemius muscle as the case may be, and the measurements that were taken. So what are the particular spinal nerves that were there? And they also found that the recovery curve of the end organ was similar to other excitable tissues, but they had longer time relations, critically describing this nature of this action potential. So they found that nerve fibers or neurons recover very quickly. So there is a refractory period and they recover very quickly, almost completely between impulses. This ensures that information transmission through impulses from the end organ through the nerve fiber happens relatively fast. It turns out that this is an all or nothing response. It is either present or it is not present. We will share with you the action potential video in which we will describe what the properties of the action potentials are. So, it is an electric response in which either it is present or it is not present, but not both. So, in other words, it is not graded, it is either present or it is not present. There is no gray area. So, with this we come to the end of this video. In this video we looked at how nerve impulses were being recorded in the good old days using electromechanical apparatuses, Keith Lucas, Edgar Adrian and others, and how single action potentials were being recorded from extracellular recordings. These were extracellular recordings made from muscle nerves, muscle spindles, and how loading, duration and amount of stimulation, duration amount of loading changes the output, and how information was perhaps coded in the action potential trains or the nerve impulse trains that were there. In the good old days, almost in all the papers of Edgar Adrian, Adrian was the first author, not because he contributed the most, but because in the good old days in Journal of Physiology, where he primarily published, the publications were all in the order of alphabetical order, and because his last name was Adrian, he was almost always the first author. A tidbit. With this we come to the end of this lecture. Thank you very much for your attention.